Hello everyone, welcome back to AS and A-Level Biology with Dr. Demi. I am Dr. Demi, and in this video, which is what I would call a special intermission, I am going to be solving past question papers. So to keep things very simple for this first video, I have chosen the multiple choice paper from 2018, that's paper 1. And please don't look down on that. But what I want you to know is that paper 1 is one of the most difficult papers in the biology exam. And this is simply because students tend to struggle with making the right choices in this paper. So I hope that as I go through this video, I'm able to share some techniques with you that would help you to get better at choosing the right answer. I'm also going to do like a structured paper so that you can see or get some tips on how to answer questions for that kind of paper as well. But I thought it would be a good idea to start with paper 1. The good thing about paper 1 as well that many students may not be aware of is that when you practice paper 1, it actually helps you see which sections in your AS syllabus you are slacking at. So for example, when I do timed assessments with my students, after we finish the syllabus, I can always pick up from paper 1 if, for example, they don't understand chapter 7. And that's because paper 1 is structured in such a way that all the topics are aligned, like they're arranged chronologically. So chapter 1 questions come first, and then 2 goes all the way to 11. So when I see that we've arrived at the section that's chapter 7 and the students are just getting most of the answers wrong, then I'm able to tell that, oh, okay, they don't really understand chapter 7, and I can sort of flag that to them to say, listen, you either haven't revised properly or you don't understand, and we're then able to work out something. Anyway, enough of that. But if you find that that might be helpful for you, it's a good way to start, and just use it to assess which topics you're really good at and which ones you're just not that great at. All right, so I'm going to get into it. Enough of all the introductory chatter. So this is May, June 2018, paper 1, and I've picked questions at random, by the way, so I'm not solving the entire paper. I've just picked questions very randomly, and some of them I figured might be difficult for students, so it would be best if I can answer them. So let's start with this one. It says, which statements about resolution and magnification are correct? So I often tell students, whenever it comes to these kinds of questions where you have two different things and statements have to be correct for the both of them, the best thing to do is to read the statements for only one before you even move to the other one so that you don't get confused. So as I'm going to do, I'm just going to read for resolution only. A says resolution is the ability to distinguish between two separate objects that are very close together. And that is true. B says the clarity of the image formed by the microscope is resolution. That's also true. C says the number of times larger an image is compared with the real size of the object. That is not true for resolution. So we already know that C can't possibly be an option. And this we are doing without even looking at what it says for magnification. But we already know that if it's wrong for resolution, then whatever is in magnification should not matter. And then D says the power of the microscope to focus on very small objects. That's also not true. It has nothing to do with the size of the object. So we can sort of say now that we've done a 50-50 split over here, where we're basically saying C and D cannot be possibilities. The answer is either A or B. Now let's look at A. So we know that A for resolution is correct. For magnification, it says the number of times larger an image is compared with the real size of the object. That's true. And B says the power of the microscope to focus on very small objects. Again, magnification has nothing to do with focusing on small objects. It's about blowing up the size of an object, so really making it bigger. So we already know then that B cannot be the answer, and that is how we're able to pick A with confidence. And you can check this in the mark scheme for this paper if you can get it, but you would find that the answer is most likely, well, the answer should be A. Nothing else seems possible here. Now, question 2. An eyepiece gradicle has a scale with 100 divisions, and it says a stage micrometer has a scale with 50 divisions, each of which is 0.04 millimeters apart. So I often tell students, whenever you're faced with questions that have lots of numbers like this, just make sure you underline the key parts. So 50 divisions, each of which is 0.04 millimeters apart. And then it says using a times 40 objective lens, the whole length of this stage micrometer scale lines up with 15 divisions of the eyepiece gradicle. What is the actual length of the 100 division scale of the eyepiece gradicle? Now, this obviously might be a lot of information, but you can solve this very easily by just using magnification is equal to image size over actual size. So I write that as I over A. And our magnification here is times 40 objective length. But remember that when you're dealing with magnification, you are also including, especially if you're using a microscope, you're also including the eyepiece. And the eyepiece always has a magnification of times 10. So that means we have magnification of times 10 times 40, which is then equal to 400. Okay, so with that said, we know that our magnification is 400 times, and we're just going to leave that there. Now, it says that the stage micrometer is 50 divisions, each of which is 0.04 millimeters apart. And that just tells us that the stage micrometer's length in total is 50 times 0.04, and that is going to give us 2 millimeters. Okay. So 2 millimeters in length, that's the stage division. So here it then says that the whole length, so this 2 millimeters here, which is the entire length of the stage scale, fits into 15 divisions of the eyepiece gradicle. So that means 2 millimeters. I'm just going to write the bit backwards here so that it's just 15 units is equal to 2 millimeters. Okay, and then we're asked to find what the actual length would be for 100 units. And so we can simply say here 100 units would be equal to 2 divided by 15 times 100. And you would get an answer that is, let's see, that should be 13.3, okay, millimeters. All right, so 13.3 millimeters, and that sort of tells us that the answer is B. Now, for many people, this might be a little bit weird because then you start saying, okay, but then what about the magnification and why have we done it this way? Because this looks really easy, and indeed, this is the easier way to get to your answer. But let's say that you wanted to be a little bit more pedantic about how you solve this question, and you've decided, okay, no, I want to go through the whole magnification process. So I'm just going to erase what I've done here. Please pay attention because this gets a bit interesting, weirdly complicated. I'm not going to erase the part about magnification because I'm going to use that. So let's say we want to then include the magnification issue here. All right, because it says that when you magnify this thing, the stage micrometer only fits onto 15 divisions on the eyepiece scale. So what you can do here then is you can say, the stage micrometer, like we said, is 2 millimeters in length. So I'm just going to write S is equal to 2 millimeters in length, right? And so this is what we would call the actual size of the stage micrometer. But we're being told that this stage micrometer has been magnified by 400 times. So we can actually say to get the image size, because it's the image of the magnified part of the stage micrometer that's now being fitted onto what's called 15 divisions of the eyepiece scale, if you want to look at it that way. So you can then say, okay, in that case, 400 times, so I'm using I over here, A now, 400 times being our magnification here would be the image over 2 millimeters, okay? And that then tells me that the image size for the stage piece graticle is 800 millimeters. This is a much longer method that takes you to the exact same answer, but I would say if you're a student listening, you don't have a lot of time, but I'm just expanding this for those who might just want to see it. And then it says here that 15 divisions of the eyepiece graticle is equal to this image length, because we're now dealing with it at a magnified scale. So 15 divisions would be equal to 800 millimeters over there, right? And so that means one division is basically 800 divided by 15, and then 100 divisions would be equal to 800 divided by 15 times 100. And if we calculate that, so I'm just going to grab a calculator over here. We will get 5,333 millimeters. Okay, So 533 millimeters. This is now, so over here, we're still dealing with the image. We don't have the actual. We're dealing with the image because we've incorporated the magnification that we were given. So we then need to go back. If this is the image length of 100 divisions of the eyepiece radical, we then need to find the actual length, which again takes us back to m is equal to I over a, and m still remains 400 times, okay? And that means I would be the image, because now we're trying to find the actual one, so I would be 5,333 divided by a, which is our variable. And over here, if we divide 5,333 by 400, we arrive at the exact same answer, which is 13.3 millimeters. Okay, and so that then still leads us to B. So the answer here is B. You can have a look in the mark scheme again if you're not sure, if it seems confusing, or just watch this again or ask questions. But I just thought to do this for students who might want to use the magnification aspects of things. So think about it again. We start with m is equal to I over a. We have a magnification of 400 times because we are dealing with 40 times objective as well as the 10 times from the eyepiece, all right? So that tells us that magnification is 400 times. The actual length of our stage radical is 2 millimeters, which we get by multiplying 50 divisions by 0.04 millimeters. And so the image size in that regard would be 800 millimeters. All right, and if 800 millimeters fits into 15 divisions on the eyepiece radical, then 100 divisions on the eyepiece radical would simply be 5,303 millimeters, 333 millimeters rather. And from there we can then go again and calculate the actual length of 100 divisions, which would lead us to 13.3. I know that's a bit long. It's a longer way. You only have 1.5 minutes per question in paper 1. But if you know you're quick and you're quick thinking, then you can just use this. It's also the foolproof way to calculate, because doing it the easy way that I showed you earlier might be wrong in certain instances. Okay, let's move on to something a bit more fun, something you probably want to do. This is question 4, and it says here, a scientist carried out an experiment to separate cell structures in animal cells. The cells were broken open to release the structures, and the extract was filtered into a centrifuge and spun in the centrifuge. And so it says the heaviest structure sank to the bottom as pellet number 1. And then pellet number 1 was removed, and pellet number 2 came through, which was the second largest structure. And so it goes on and on like this. And the point of this question is simply to ask you, what would be the heaviest of all the cell structures that you're given? So you're given things like nucleus, lysosomes, mitochondria, ribosomes. So just first of all, pellet 1 is supposed to be the heaviest. So when you're here, the first thing I would say is look at pellet number 1. It's supposed to be the heaviest. Now, the places where you're seeing ribosomes, for example, those cannot possibly be the heaviest. They are so tiny. So that already tells you that you can eliminate C and D, because pellet number 1 cannot possibly be ribosomes. It's supposed to be the heaviest. It can't, ribosomes, for example, cannot be heavier than the nucleus. So that tells you that C and D are not options. So you've already cut your thing, whatever you're paying attention to just 50% so that you're able to zoom in better. You'll cut your choices down to 50% so you can zoom in better. So now let's look at A and B. So A says the nucleus is pellet number 1, and then it says the lysosomes is 2, and then the mitochondria is 3. Now lysosomes are not heavier than the mitochondria. The mitochondria, remember, has its own DNA, has a whole lot of structures that are inside of it. It's basically like an independent cell on its own. So this sort of tells us already that we can't have option A as the answer. The thing that we're left with then is option B, which tells us that the nucleus, the mitochondria, lysosomes, and the ribosomes being the smallest. That would be the answer. The reason why the nucleus is so small, is so big rather, or heavy, not big, is that it has the nucleolus inside of it. It also contains the entire DNA, and also has some ribosomes, I believe, in the nucleolus. So that tells you that it contains structures within it that would then make it heavier than the other structures. So that is how you would answer a question like this. I want us to move a bit swiftly, but I do understand the importance of making this video as valuable as possible to you, so I'll try not to be too quick. So let's look at number 5. Radioactively labeled nucleotides are introduced into a cell. And it says, in which cell structures will the radioactivity first become concentrated? So if you look here, we have 1, which is pointing at the rough endoplasmic reticulum. And we have 2, which is the nucleus, we have 3, which is the mitochondria, and then we have 4, which is the Golgi body. Now remember that the Golgi body is involved in the folding and modification of proteins, okay? So you're thinking, okay, well, maybe not, but if you look at this, this is an actively secreting cell. So I would go ahead and eliminate the Golgi body as a possibility, because this suggests when we're talking about nucleotides, we are probably thinking about something that has to do with DNA or RNA. And so that just tells me that the Golgi body would not have anything to do with it, because we're not building proteins. So let's then look at what is left. For the rough endoplasmic reticulum, again, that is a protein building structure on its own because it has ribosomes on it. So that I would say is not an option. And if we then go through, let's see the options that have 1 and 4. That would be A, so A is not a possibility. B is not a possibility. D also can't be a possibility. We're left with 2 and 3. And 2, simply because the DNA is in the nucleus. So if you have radioactively labeled nucleotides, you would most likely see them in the DNA. And you would see them also in the mitochondria, because the mitochondria also has DNA. And so for that reason, the nucleotides, because nucleotides are used to assemble they're used to assemble our DNA and RNA, so that's where you typically find them. So I would go ahead and say that the answer to that one is C. Okay. Number 6, what is the general formula for amylose? Now, this is another question that I think confuses students a lot, because students typically know that amylose is made up of glucose, alpha glucose to be specific, and the formula for glucose is C6H12O6. So many students are often tempted to pick D as the answer, but the answer is not D. The answer is C, and I'll tell you why. Now remember that when you're making glucose and when you're making amylose, for example, you need to add many repeating units of alpha glucose together. And when you do that, if you remember from chapter 2, what that means is that you are carrying out condensation reactions. So one alpha glucose plus another alpha glucose is a condensation reaction that releases water. That water means that H2O is deducted from the glucose molecule, or the glucose, the disaccharide. So that means that when you have many repeating units of glucose, they all don't have the complete structure of glucose. They've all lost water. And so in that regard, you simply need to look at the formula for glucose and deduct H2O. If you deduct 2 hydrogens from here, you're going to be left with H10. 1 oxygen, you're left with H5. The answer here is C. That's how you solve that question. Let's move on to the next one. Okay, so let's see this one in number 8. It says here, in unsaturated lipid molecules, where are double bonds located? This one is very important. So many students often make the error and they say between fatty acids and glycerol. That's not true. So in your unsaturated lipids, so this is typically what they would look like. And this here represents a hydrocarbon chain. And this is just like from from glycerol, and this is from the carboxylic chain on the fatty acid. Your double bonds are typically located within the fatty acid chains themselves. You don't count this bond over here as part of the double bond. This is part of the carboxylic group. So you don't necessarily count it as one of the double bonds. You only count the ones within the hydrocarbon chain. So the answer here should be C, within fatty acids only. Okay, let's look at number 9. Number 9 says here, phospholipids and triglycerides are important biological molecules. Which properties are correct for these molecules? Again, remember what I said to you in the first question. I said just read for one part, and then you can see what other part would follow. So phospholipids, are they nonpolar? They are not nonpolar. They are they are polar molecules. Actually, they have polar heads. So that already tells us that A and B cannot be correct. All right, because here it says non-polar phospholipid, so that already kicks them out. Now we are left with C and D, and we can just sort of zoom in there. Partially hydrophobic, so non-polar, triglycerides are certainly non-polar. Partially hydrophobic, that's a phospholipid, that's for sure. Partially hydrophobic, that's not true for triglycerides. They are fully hydrophobic. They don't want anything to do with water. And so that leaves us with C as the only possible answer. And so we know C is correct. Let's look at number 10. I hope you are following and you're not feeling overwhelmed by these questions. And I hope you're also like trying to guess the answers before I solve the question, because that would also help you just to see how well you can do. So let's look at number 10. Number 10 says the diagram shows two arrangements of amino acids in a protein. Which row correctly names the bonds at S or T? So first thing I would say when you have a question like this is identify these two structures. What exactly are these? It says amino acids in a protein. So you know this is some form of protein structure. This is certainly not a primary structure because the primary structure is simply the sequence of amino acids in a polypeptide chain. This looks like an alpha helix to me. Over here, this looks like an alpha helix, and this over here looks like a beta pleated sheet, and these two are secondary structures of proteins. So you know you're dealing with secondary structures, and that way you can sort of ask yourself, okay, well, which bond is typical in a secondary structure? And what the bond that's typical, obviously the peptide bond between the amino acids themselves in this helical chain, but also these bonds here where you have S and T, those are hydrogen bonds, right? Hydrogen bonds are found in the secondary structure. They're the only bonds besides the peptide bonds in the secondary structure of a protein. So already we've solved this because everywhere where it says T only or absent, we know that B and C can't be correct. Ionic bonds do not exist at all, so D can't be correct. A has to be our correct answer. And by the way, guys, you can check out these in the mark scheme if you feel like I'm doing them wrong, but I am very sure that these answers are correct. Let's look at number 13. Number 13 says here that the effect of substrate concentration on an enzyme catalyzed reaction was measured in 3 different conditions: with no inhibitor, with a competitive inhibitor, and with a non-competitive inhibitor. And I hope you guys remember inhibition and how that works. And so we have the graph that shows the results, and it says which statement is correct. So statement A says X is a competitive inhibitor which binds to a site other than the active site of the enzyme. That is certainly wrong. So already just reading that, you can tell it is wrong. The reason this is wrong is that competitive inhibitors bind exactly to the active site of the enzyme. So X can't be a competitive inhibitor and bind elsewhere. And then X is a non-competitive inhibitor which has a similar shape to the active site of the enzyme. That again is wrong. A non-competitive inhibitor does not work with the active site of the enzyme. It binds at the allosteric site. So just to do an illustration here for you, this is usually the illustration I use in class. So if this is the active site of the enzyme over here, a competitive inhibitor would bind exactly at the active site, while a non-competitive inhibitor would bind here, which would be called the allosteric site. And by binding at the allosteric site, it changes the shape of the enzyme, and then the enzyme is unable to bind to its substrate. So A and B cannot be correct. C says Y is a competitive inhibitor that has a similar shape to the active site of the enzyme. Now that would be correct. That's the correct definition for a competitive inhibitor, a similar shape to the active site. But if you look at the graph for Y, Y certainly is a non-competitive inhibitor based on how much it has reduced the rate of the reaction. Non-competitive inhibitors, because they are able to change the shape of the active site when they bind to the allosteric site, they just mess things up over here, so the substrate is unable to bind. Competitive inhibitors, on the other hand, they would also bind. So let me just erase this over here. that was an error, but I'll just draw it again. Competitive inhibitors typically can bind to the active site of the enzyme. Let's assume this is the active site. But if you increase the substrate concentration, the enzyme would still be able to carry on the reaction. So the reaction would be a lot slower. So inhibitor X would be a competitive inhibitor. The reaction would be a lot slower, but it would still happen as substrate concentration increases. Whereas with non-competitive inhibitors, because they've changed the shape of the active site, no matter how much you increase the substrate concentration, the substrate just is unable to bind. So that tells us that C can't be correct because Y is certainly a non-competitive inhibitor. And D says Y is a non-competitive inhibitor which binds to a site other than the active site of the enzyme. And so that is our answer, D. Okay, let's go very quickly here. So number 14, which statements about cell surface membrane are correct? Channel proteins allow water-soluble ions and molecules across the membrane. That is certainly correct. 2 says glucose can pass into the cell via carrier proteins. As a matter of fact, that is the way that it goes into the cell. Oxygen passes freely through the membrane as it is soluble in lipids. That is also correct for oxygen. It can pass through the membrane because it's soluble in lipids. Some glycoproteins act as antigens. That is correct. This is a chapter 4 question. So already it's just asking you about the functions of the different components of the cell membrane. So because all of them are correct, there's no reason for us to labor or agonize over the different options. Just look for where everything is correct. 1, 2, 3, and 4, you're done. That's A. Okay, I hope this is helpful as we're going along. And then let's look at number 15. Which of these features increase the efficiency of ion uptake by a root hair cell? And so it says here, option 1, many mitochondria in the cell, 2, high concentrations of ions in the vacuole, and 3, protein carriers in the cell surface membrane. Now, when you look at these statements, I often tell students, whenever you're giving these 1, 2, 3 statements, just look out for one that's true, which one's true and which one's not true. That's often the best way to start so that you don't agonize by saying, okay, A says 1, 2, 3, let me read 1, 2, 3. And then B says 1 and 3, could it be that? Don't do that. Rather, just start with what you know. So ion efficiency uptake by a root hair cell could be a result of active transport if you have a high concentration inside the root hair anyway. So 1 could be correct, okay? So that's 1. High concentration of ions in the vacuole would not necessarily increase the efficiency. So here, let's focus on what the question says. What increases the efficiency, all right? So high concentration of ions in the vacuoles, that has no impact whatsoever on efficiency. Protein carriers in the cell surface membrane, certainly they would help. And so based on this, I would go with B for this question, that number 15 is B. And I hope that you're able to find that it is B as well. Let's look at number 16. What can increase the fluidity of the cell surface membrane at low temperatures? This is always a good one. I absolutely love this question. And I think there was a student, one of you who was watching the videos and helped to clarify the fluidity and how that affects the close packness of the cell membrane. So if you haven't watched the chapter 4 videos, please make sure you go and check them out. So here, what can increase fluidity of the cell surface membrane at low temperatures? So remember that the cell surface membrane is made up of phospholipids, and at low temperatures, lipids would typically try to solidify. And if they solidify, that affects the transport mechanisms across the membrane. So you need them to not solidify. You need things that are going to make them less closely packed. And so if you think of it that way, when you have double membranes in a fatty acid chain, that certainly makes things less closely packed because double bonds also introduce certain kinks into the fatty acid chains that then make them sort of space out a little bit. Cholesterol, this is the function of cholesterol in the cell membrane to regulate fluidity. So we can't leave cholesterol out. It certainly has to be there. And 3 fatty acids having shorter chains, that's also going to help with them not being closely packed and being more fluid. So the answer here would be A. All right. And from here we skip to 23. Like I said, I picked questions quite randomly. So if there's a question that you have in mind that you just feel like can't help, you can take a picture of it and try to upload it in the comments and let everyone just jump in and try to help you, because I think that would be great. Or you can just type it out if you're able to. Okay, which is the correct DNA triplet on the original DNA template that codes for the amino acid histidine? So here, histidine is GUA. All right, and what they're telling us to do here is to work backwards. So if you watch the videos on protein synthesis, you remember that you have DNA to mRNA, and then to tRNA that binds with the mRNA, and then you have your protein being made. Okay, so the tRNA carries the anticodons. All right, it brings the anticodons. The mRNA is made from the codons. So if it's telling us here that this is the anticodon GUA, then it means that the codon for GUA, remember G binds to C, U binds to A, and A binds to U. So it's telling us here that the codon is CAU. Now, if this is CAU in the mRNA, then what is the DNA? That is what this question is asking us. What is the DNA? DNA, C would bind to G, A would bind to T, and U would bind to A. And so the answer here is C. All right. Now let's look at 26. Which features of companion cells are essential to their function? Again, just like I said, whenever you have questions like this, start with what you know. Don't try to be, oh, I'm going to read option A and then go look at the sentences, and option B, look at the sentences again, because that would make you confused. So let's start here. They are connected by a plasmodist matter to the sieve tube elements. That is certainly correct, that the companion cells are connected by plasmodist matter, so the sieve tube elements that are used to transport sucrose. And then 2 says they have a thinner wall than a sieve tube element. That's actually not true. The companion cells are actually, they're metabolically active and quite strong cells. They have a nucleus and mitochondria. That is true. Just like I said, they are metabolically active, and that is how they're able to aid in the transport of sucrose. And so that tells us then that only option C could be possible, because all the others 2, they have 2 in them, and so 2 is not an option. All right, let's look at this one. I found this one quite interesting. It says here that the diagram shows the changes in electric activity in the heart muscle during one cardiac cycle, and this is called the electrocardiograph. And it says which electrocardiogram shows a rate of 75 beats per minute. Now, this is not hard. I just have to say that it's not hard, and I know that students typically just go, huh, what exactly is going on here? How am I supposed to know this? So remember, when you're looking at these options here, that from here to where you have the next, so with this, I'm just going to circle it. This over here is 1 heartbeat. Okay, so you can just count the peaks if you want. So you can just say 1, 2, 3, and this is 0 to 1. That's 1 second, 2 seconds, 3 seconds, just 4 seconds over here. So if you have 3 heartbeats in 4 seconds, then in 60 seconds. So 3 heartbeats, 4 seconds, and 60 seconds, if you multiply that, it should be by 15, you would only have 45. So that already makes it impossible that A would be your answer. Let's look at B. Here we have 1, 2, 3, 4, 5. So we have 5 beats in 4 seconds. So I'm just going to write 4S is equal to 5 beats over here. So that means in 1 second, it's just 5 or 4 more or less. And then in 60 seconds, which makes up 1 minute, you would have 5 divided by 4 times 60. And 4 in 60 is 15. And 5 times 15 is 75. And so that tells us that the one that's giving us 75 bits per minute has to be B. If you have some time, you can go ahead and check for C and D, but I would tell you that they're certainly wrong just by counting the peaks. Like over here, this is 1, 2, 3, 4, 5, 6. If you have 6 bits in 4 seconds, there's no way you would get 75 bits in 1 minute. And the same goes for C, which is obviously a very fast heartbeat over there. Okay, and this is the last question I chose in case you're beginning to feel like, oh my goodness, when does this end? So this question here, again, I chose it because it has the 1, 2, 3, 4 thing where you have to read through sentences. And again, please just, as I'm speaking, you can start reading the question on your own, but just try as much as possible to stick with what you know before you even start to read the options, so that way you're able to choose properly. So here it says some animals have genes that code for small peptides called cathelicidins. Well, I hope I said that correctly. These peptides kill a wide range of bacteria by attaching to lipids in bacterial membranes and weakening and disrupting them. Scientists have produced a synthetic version of this peptide that kills bacteria, a synthetic version of the peptide that kills bacteria that are resistant to a number of antibiotics such as tetracycline. So basically we're using this peptide to kill antibiotic resistant bacteria. Which pair of statements, so now it says which pair, so you're looking for two statements, just two statements that are correct. Which pair of statements explain how the synthetic cathelicidin, or cathelicidin would be correct, might help with the problem of antibiotic resistance? First one says here, it is synthetic, so bacteria can never become resistant to it. That is not true. Bacteria typically would develop resistance once they understand the mode of action of anything that attacks them. So that cannot be possible. The fact that it's synthetic doesn't mean that bacteria would not become resistant. 2 says it can be used instead of tetracycline, allowing tetracycline resistance to be reduced. That is correct. Now remember that the more we use antibiotics, the more resistant the bacteria become. So if we reduce our use of antibiotics and we use synthetic molecules like cathelicidin, then we might be lucky and have tetracycline resistance reduced. Also because bacteria pass resistance to one another. So 2 is certainly a possibility. And then 3 says, the only way a bacterium could develop resistance to it is by altering all the lipids in its membrane. That's not true. So bacteria can develop resistance in a variety of ways. So even though it attacks the lipids in the bacterial membranes, the bacteria doesn't have to change every single lipid in its membrane in order to become resistant. Just by changing maybe just a couple of carbon chains or something, that might result in some form of resistance. So 3 can't be correct. And because we are looking for 2 statements, we have hope that 4 would be our second statement that's accurate. It could be used to kill multi-drug resistant strains of bacteria for which there is no effective antibiotic. That is true. Whenever you have multi-drug resistant strains of bacteria and they're unable, like no antibiotic is able to fight against them, sometimes synthetic molecules like this, because the bacteria are not yet aware of its mode of action or they haven't developed resistance, would then be helpful. And so I would go with the answer D as the possibility over here. So yeah, that brings me to the end of this question answering session. And I hope that you have found this helpful, that it contains techniques that are helpful for you. If you have any questions, again, this is the 2018 paper 1 paper. So if there are questions from this paper that you feel, okay, well, I could use some help with this particular question, if you're able to take a snapshot and just put it there on the in the comments, or just type it out so that your colleagues can also jump in and help you, because I can't promise that I'll always be able to help, but when I can, I certainly will respond. But if you're able to just take a snapshot or type it out, please put it in there so that your colleagues are also able to help, and you can sort of develop a learning community together that I think would be very helpful for you. So that brings me to the end. Thank you so much for watching. I am working on preparing the slides for chapter 14, which is homeostasis. But before then, I'm going to upload some questions from paper 2, also 2018, May, June, so you can get some tips on how to answer paper 2 questions properly. All right, guys, have a good time. Until the next video, goodbye.